So can we start now?
And doctor.
Okay. So my lecture today is host-pathogen interaction. My name is Dr. Nik Hazrul. I'm from the Department of Medical Microbiology and Parasitology, USM. So the contents of my lecture is introduction, types of host, types of pathogen, types of interaction, the factors affecting the host, the factors affecting the pathogen, and the pathogen and pathogenicity and virulence, pathogenesis and component of infection, transmission. So, as we all know, you are not alone in this world. There are diversity of living organisms, methods of obtaining food, and habitats, both macro environment and micro environment. So each organism are adapted to the environment they are living in. So like fish, they are adapted to live in the water, and humans are adapted to live on the land, and birds are adapted to able to fly. So also the microorganisms, the tiny microorganisms, they are also adapted to survive in different environment in the host. So most infections are caused by only a few organisms. Although there are so many microorganisms, only a few causes disease. And normal individuals harbor a large number of microbes. We call them as normal flora. So these normal flora are beneficial. So there are a lot of microorganisms living in the host, in ourselves as the host. And these are beneficial. They don't cause disease. So only a few causes disease. So how do we determine which of the microorganisms we get from your sample, which one is the one causing the disease? Whether we use the Koch postulate. Koch postulate says that the organism should only be found in the sick animal, not in the healthy animal. The organism must be able to be cultivated in a pure culture. When inoculated in susceptible animal, it must produce the same disease. And the organism should be able to be re-isolated and recultured generation to generation. So this is an illustration of the Koch postulate. The microorganisms are isolated from a dead animal, and then they are able to grow the organism in a pure culture, and the microorganisms are identified in the lab, and then it can be injected into a healthy animal, and when injected into a healthy animal, it produces the disease. And this uh, organism can be again re-isolated in pure culture and identified again. So this is what we do in the lab. When you send blood culture, you send urine sample, we do this in the lab. So that's how we know that that organism is the one causing disease in your patient. So that's the role of the lab. So let's go to definition. The host is an organism that harbors a gas, the gas typically being provided with nourishment and shelter. So the host provides this nourishment and shelter to the gas, the organism. And pathogen is organisms which are capable of causing harm and damage to the host. So the pathogen is the organism which cause disease. We call pathogen. And infection is the growth of the organism in a host. So host-pathogen interaction can be both beneficial, like I said, like the normal flora, and it can be harmful, like the pathogen. Or it can be neutral, depending on factor or the condition. Like in an immunocompromised host, then it can be pathogen, called as opportunistic pathogen. So, for introduction, the host body provides a favorable environment for many microorganisms. It is rich in nutrients and growth factors, and different sites or organs confer different growth niche for different organisms, like the skin, respiratory tract, gastrointestinal tract, or oral cavity. And all are different niche, which are suitable for different organisms. Colonized by normal flora, essential for maintaining healthy condition. So the microorganisms we call a normal flora, they are very beneficial for us. We need to have them in our body to maintain a healthy condition. So the pathogens have same objective. All pathogens, so many pathogens, but all have same objective. They have to invade the host, then they have to evade the immune system, then they establish their colonies, and then they transmit to other hosts. So this is accomplished based upon their interaction with the host. The host-pathogen interaction is a complex ongoing battle between the pathogen's virulence strategies and the host defense. So both have their own properties. Pathogens invade and colonize the host tissues, and the host block the invasion and destroy the pathogen. So this is an interaction between the pathogen and the host. The pathogen want to survive, they invade, they establish their colonies, and the hosts have to block this invasion, and they have to destroy the invading pathogens. So frequently, the host immune response to the pathogen interaction is responsible for the symptom and disease in the host. You get the symptom and signs you get is because of this interaction, the host immune response to the pathogen. So when we describe the organism, we use two words. One is pathogenicity, one is virulence. So it may seem similar, but you need to understand what we say when we say pathogenicity. Pathogenicity is the ability to cause the disease. So whether it's pathogenic, non-pathogenic. So for example, non-pathogenic, most nocardia species are non-pathogenic. Only so many, there are almost 200 nocardia species, and most of them are non-pathogenic. But a certain few species from the nocardia genus, Nocardia asteroidis complex, is a pathogen. That's how we talk about organisms. So the vast majority are harmless, non-pathogenic, and relatively small list, like this few species, are pathogenic. And when we talk about virulence, we talk about its severity. So an organism we say is low virulent or it is a high virulent. For example, Staphylococcus aureus is a highly virulent organism. When we get Staphylococcus aureus from a patient sample, we immediately have to treat. There's no question about it. We have to treat because it's a highly virulent organism. It causes a lot of damage to the host. But when we get from the patient sample, we get coagulase negative staphylococci such as Staphylococcus epidermidis, then you don't have to treat because it's a low virulent organism. Most of the time, you don't have to treat, except for certain condition like immunocompromised hosts, patients on oncology patient, on oncolytic treatment, then you need to treat. But most of the time, cons is a contaminant from the skin, Staphylococcus epidermidis, which do not need to treat. Now we go to the types of hosts. So types of hosts can be reservoir, predilected, definitive, secondary, or paratenic. So a reservoir host is an organism that harbors a pathogen, but the host suffers no ill effects. So the host may not even realize that it is a host, but it keeps on spreading to others because it does not realize it has an infection. So they are the source. If an outbreak or anything, they are the source. For example, like COVID-19 is the bats. So the virus survive in the bats without causing any damage to them, but when they spread to pangolin and pangolin to human, and then the humans get disease COVID-19 and causes an outbreak. And then there is a predilected host. So the predilected host is a preferred host. For example, with Salmonella typhi, it does not cause disease in other animals. It causes disease in humans. So humans are the predilected host for Salmonella typhi. And then there is definitive or primary host. Primary host or definitive host is an organism which the parasite reaches maturity and reproduces sexually. So when you see the cycle, the life cycle of an parasite, mostly parasite, then you see in which organism there is the parasite reach sexual maturity. So in that host, we call that as a definitive host. And then there is a secondary or intermediate host, is where the organism harbor the sexually immature parasite and is required by the parasite to undergo development and completes its life cycle. For example, the pig ingests from eggs in human feces. So the pigs is the secondary host. And then there is a paratenic host, which is an organism that harbors the sexually immature parasite, but it's not necessary for the parasite's development cycle to progress. For example, humans and horses are dead-end hosts for a West Nile virus, whose life cycle is normally between culex mosquito and birds. So the humans and horses are dead-end hosts for the West Nile virus because it's not usually part of the life cycle. The life cycle is usually between the mosquito and the birds, but the humans and horses are the end hosts. So we call humans and horses as a paratenic host. So this is the life cycle for the tapeworm. Tapeworm. So the human ingests the meat from the pig, the pork. So the parasite develop into the mature stage in the humans. So the humans are the definitive host, and the parasite, the tapeworm, grow in the pig. The pig is the secondary host because in the pig it does not reach maturity. It doesn't reach sexual maturity. So it cannot reach the sexual stage in the pig. It reaches sexual stage in the human. And then again, we talk about the type of host. So the reservoir, like we said just now, is the best. It's the source of the pathogen. It can be both also living and non-living. So reservoir can be also non-living, such as in the hospital environment, the surface can be a source for outbreak also. There can be organism on the surface. That's why you need to always use the alcohol wipe on the use on the chlorine on the floor, because they can become a source for infection for the patient in ICU. Can be a source, so it can be a reservoir, like also, and also for the living, like humans, animal we described just now. And then there is also a few types of carrier: the passive carrier, incubating carrier, convalescent carrier, and also the active carrier. So the carrier host that harbors the pathogen. They are asymptomatic, subclinical, or non-lethal infection. So they can be passive carrier, carry the pathogen without ever having the disease. So the passive carrier is similar to the reservoir. They carry the pathogen without ever having the disease. And then there is an incubatory carrier. It's capable of transmitting the pathogen during the incubation phase. So in the cycle, in the life cycle, we will see later the incubation phase. This is during the window period. So during the window period, in the incubation phase, it can spread to others. We call this type of host as a incubatory carrier. And then there is also a convalescent carrier, which is capable of transmitting the pathogen during recovery phase. So in the life cycle again, we will see later the life cycle. So when it's transmitted during the convalescence phase, so this type of host we call convalescence carrier. And then there is an active carrier which have completely recovered from the disease but continue to harbor the pathogen. Some people, like they have Salmonella typhi, even treated, they completely recovered from the disease, but still they harbor the pathogen, the Salmonella typhi in their gallbladder. They can be persistently spread to others, like in the history, like typhoid Mary. They persistently transmit the disease to others, even after recovery. Next, we go to type of pathogen. So pathogen, there are obligate pathogen, opportunistic pathogen. So obligate pathogen, pathogen that cannot survive in any manner outside the host, and there is an opportunistic pathogen, pathogen that may exist in a free living state in the environment for some time. Okay, obligate, cannot survive in any manner outside of the host, and opportunistic, they may exist in a free living state in the environment. Next type of parasite, there is ectoparasite and endoparasite. Ectoparasite exists on the surface of the host like mites and ticks. They can, in the hair, you get ticks. So they are outside surface of the host. And then there are endoparasites which survive in the digestive tract like amoeba and worms. Some survive in the genitourinary tract like ciliates and flagellates. There are also endoparasites which survive in the hepatobiliary tract like Clonococcus sinensis, liver flukes, and there are parasites which survive in the blood and tissues like Plasmodium causing malaria and filaria organism. They can survive in the lymphatic tracts, and this one in the blood. So when they survive in the host, they are called endoparasites. So next, we know the type of host, type of pathogen. Now we go to the type of interaction. So the interaction can be some symbiosis where the organisms live together. There can be mutualism where their existence benefit both the organism and the host. It can be commensalism where it benefits the organism but does nothing to the host, such as the normal flora. And it can also be parasitism where the organism drives all the benefit and harm the host. And there can also opportunistic infection where the commensal exploit circumstances of the host like immunocompromised host to become pathogen, such as Candida albicans in the HIV patient. Because HIV patients are immunocompromised, in that situation, it causes the damage to the host. But in normal situation, they do not cause damage. In small amount, they do not cause damage. And also, it's a Acinetobacter baumannii. It's a low virulent organism. It's everywhere in the environment. And in normal host, the normal host immunity can defend itself. But in immunocompromised, the immune system is weak. Even low virulent organism can cause infection. We call as nosocomial infection. In the hospital environment, we call as a nosocomial infection. Usually in ICU patient immunocompromised, it can cause opportunistic infection. So what are the factors which affect the host? There are specific factors and non-specific factors. Specific factors are directed against specific species or strains of pathogen or immune system. And there are non-specific factors like the normal flora, which produce bacteriocins, and they compete for nutrients. So these factors affect the host. Whether the host situation, they have normal flora in their system, or the situation is normal flora has been killed, maybe by broad-spectrum antibiotic. These factors affect the host. And then there are also anatomical physical barriers like skin, mucous, cilia. So when the patient have a cut, they have a wound, the pathogen can penetrate the barrier, the skin, and enter the host. Some like chemical barrier, like the gastric acid in the stomach, we have gastric acid. This will kill the organism, enter through our digestive system, protected by gastric acid. But if patient taking, they have duodenal ulcer, they take treatment for the gastric ulcer, and the acid level in their stomach is low, so the organism can enter. So because the gastric acid is the protection, but if we take treatment for the duodenal ulcer, acid level is low, then organism can enter the host. We also, our ears have earwax, and also in the blood there is iron binding transference. So also we have reflex nose. If organism enter our nose, we sneeze, and if organism enter our airway, we cough. This is our reflex. So these are non-specific factors to protect ourselves. And then there are also host specificity, host tropism. Salmonella typhi only infects humans. This is a predicted host. So it's specific. Then there are also tissue specificity, tissue tropism. So certain site of infection exists of binding site like Clostridium tetani. The tissue is potent through the wound, but not if ingested, because it's tissue specific. Then there is also genetic defect, like in some people they have a sickle cell anemia. So Duffy blood group with malaria, and they have a Duffy blood group. So this protect from malaria, Duffy blood group. So in African population, they have this Duffy blood group. So the incidence of malaria is lower among them. And then there is also immune status, so immunocompetency, immunocompromise, susceptibility, patient on oncologic treatment, patient on organ transplant, HIV. All these are factors which affect the host. And also there are also host conditions such as age, stress, and diet. These also factors which affect the host. And then there are also factors which affect the pathogen. So the pathogenicity of the organism, whether the organism is a commensal or opportunistic pathogen, and the ability to survive. So whether the organism can produce cyst or spore, whether they have various structures, and whether they have developmental stages to be able to survive in the host. And also whether they have the ability to invade or attach and adhere and penetrate the tissue cells. So all these are factors which affect the pathogen. Next is the ability to evade or resist immune response and defense mechanism. So once they invade the host, they need to be able to resist the host immunity. So they have certain properties like bacteria, they have a cell wall. Some bacteria like Streptococcus pneumoniae, they have capsule. Some parasites have strong cuticle. Tapeworm have suckers and hooks. So they all have evasion strategies, and they select for certain sites, tissue to localize in the host. And there are also ability to obtain nutrient and metabolite with special organs and production of enzymes. Specialized teguments in tapeworm, they have specialized tegument to attach and obtain nutrient. They have iron, certain bacteria have iron extracting siderophore to obtain iron from the host. And then some have the ability to colonize and reproduce. So bacteria, they reproduce by binary fission, asexually, and produce large amount of eggs. Certain parasites, they produce large amount of eggs, and some parasites, they are hermaphrodite. So they have both sexual organs to reproduce. So this helps them to survive and invade the host. And they also the ability to be successfully transmitted. So it helps them to their transmission, such as special developmental stage, which exploits the host environment and natural mechanism. Like worms, they have different life stages in their life cycle. The filaria, they have periodic circulation. So filaria, they survive in the lymphatic system of the human. So during daytime, they are in the lymphatics. Only nighttime, they are in the blood. So they have periodic circulation. So if you take blood sample, you need to take at night. You cannot take the blood sample at daytime because of this. Then there are also other organisms like typhoid and cholera epidemic, which spreads transmission is higher during floods. So countries which have a lot of floods, they have a lot of typhoid and cholera because of the how the organisms are transmitted during because of the water. They contaminated the water. And then there are also organisms like TB, which like they spread through dust, fomites, and water particles. So this helps them with their transmission. So again, virulence is a relative quantitative degree of pathogenicity of a pathogen to cause disease to a host. So like we discussed just now, virulence of organism can be a high virulent or low virulent organism. And then we talk about evasiveness and toxigenicity. So some organisms, they are highly toxigenic. They produce very toxic substance. But they themselves, the organism themselves, they are weakly invasive. So they cause damage because of their toxin. So their toxin, the substance they produce, can cause paralysis and death. But they themselves do not cause the damage. The toxin cause the damage. Whereas some organisms, like Streptococcus pneumonia, they are weakly toxigenic but highly invasive. So the toxin they produce are weakly potent toxin, but they themselves they are very invasive. They cause the lung damage and death. So virulence can be measured or estimated because we say just now there is a low virulent and high virulent. So we use this scale, poison scale. So lethal dose 50 is the lethal dose required to kill half the members of a tested population after a specific test duration. So water is safe and botulinum toxin is the most dangerous. So we use this scale. And then there is also infectious disease 50. So we describe the organism infectiousness using this measure. So ID 50 is infectious dose for 50% of the host, the number of pathogen that will cause a demonstrable infection in 50% of inoculated test animal. So when we say Salmonella infection, Salmonella species has a high ID 50, meaning if they ingest a small organism, they are still their host immune system still able to resist. But if they ingest too much, 1 million organism, then the host becomes sick. They don't get sick because of a small amount of organism they ingest. If they ingest too much, then they can get sick. Whereas Shigella, even if you ingest small amount of organism, because Shigella species is a low ID50 organism, even if you ingest small amount, you already get sick. So that's the how we describe the difference between organism. What's both cause diarrhea, both spread through contaminated water, but they are different in terms of their ID50. And virulence can be inherited and transferred, so it can be acquired or inherited. Like in the bacteria, they have plasmid, phage, and transposon. So some bacteria they have this Bales, aggressins, and Roslin virulence. So it's substance extracted from the virulent pneumococci, which change non-virulent to non-virulent pneumococci into virulent through this substance, virulence. So virulence genes on mobile extra-chromosomal genetic material, recombination into a chromosome, and conserve as a chromosomal gene. So they can acquire through this extra-chromosomal genetic material. And there are also through pathogenicity islands, like this is inherited chromosomally, the chromosome. It's originate from gene transfer and large organized groups of genes, 10 to 200 kilobases. So some are plasmid encoded and phage encoded, like E. coli. I have heat labile and heat stable enterotoxin, E. coli and Shigella. The adherence factors and gene products involved in the mucosa infection, and Bacillus anthrax. They have capsule production, edema factors, and lethal factors. And some are phage encoded. Phage are virus, bacteriophage. So Clostridium botulinum, they produce botulinum toxin that causes paralysis. Corynebacterium diphtheria, they have diphtheria toxin that inhibit human protein synthesis, causing diphtheria. And there is also Vibrio cholera, which cholera toxin that causes severe watery diarrhea. So the virulence factors encoded by genes on mobile genetic elements are plasmid and phage. So this can be transferred. And then for the pathogenicity islands, E. coli, they have Pi1, which produce alpha hemolysin, femre, pilu additions in UTI. E. coli EHEC, O1A7, they have macrophage toxin of the enterohemorrhagic E. coli EHEC. Salmonella typhimurium, they have SP1, invasion and damage of host cell and diarrhea, Yersinia pestis, causing the plague. They have HPI, PGM, enhanced iron uptake. Staphylococcus aureus, they have SCC-MAC, methicillin, other antibiotic resistance. The MRSA, so Staphylococcus aureus, which has this SCC-MAC, they are resistant to vancomycin. And Staphylococcus aureus that have SAPI1, they produce toxic shock syndrome, toxin 1, enterotoxin. So these Staphylococcus aureus are usually you can treat with Coxa, but once they have this pathogenicity island, they become resistant to the antibiotic cloxacillin, and you need to treat with vancomycin. And some Staphylococcus aureus, they acquire this pathogenicity LSAP1, and they produce the toxic shock syndrome. And some Vibrio cholera, L-TOR01, they acquire this VPI1, they produce neuraminidase utilization of amino acids. So virulence can be regulated, adaptation to the macro environment and the micro environment. There is complex signal transduction, temperature, pH, iron availability, osmolality, growth phase, existence of ions, and existence of nutrients. Diphtheria toxin of Corynebacterium diphtheria greatly enhanced in medium with low iron, and expression of cholera toxin is higher at pH 6, so acidic environment, temperature 30 degrees, so it grows, but they produce more toxin in this environment, in a acidic and low temperature environment compared to the alkaline normal temperature. Expression of virulence genes in Yersinia pestis is enhanced at 37 degrees and suppressed at low temperature, high concentration of magnesium sulfate. So this is their growth condition, which affect their virulence production. So the virulence can be attenuated, also can become reduced when the pathogens are kept in the lab for many generations. They are passage many generations, they lose their virulence. So lab are not an optimal growth condition, but some like the certain virus, we use this property to produce vaccine because we need the virus to lose their virulence so that we can get a low virulence virus so that we can produce the vaccine. So we passage so many generations until we get the same organism but low virulence organism and low virulence virus so that we can inject to humans so that we can protect the humans from the pathogenic virus. So this is how vaccines are, influenza vaccine are produced. So virulence is a complex interaction. It can be influenced by genetic makeup of the pathogen, genetic makeup of the host, different individual immune response, past exposure or vaccination, and environment. So next we go to pathogenesis. This is the cycle of pathogenesis of the infection. So first infection enter the host. There is a window period, incubation period, where there is no symptom, and then some have a prodromal, they feel getting a disease, but they don't have any symptom sign yet. And then they reach the acute stage where the actual disease is produced. You have all the symptom and sign, and then they recover. So there is a convalescence phase, and some transmit during this phase, and then the transmit to other hosts, and the cycle continues. So the time between the exposure to an agent and the first appearance of clinical symptoms is called the incubation period. So and the infection enter the host, no symptom yet, it's called a incubation period. So prodromal is the early non-specific, you don't have any symptom sign yet. The acute stage is where you have the onset of all the sign and symptoms. And then there is also recovery convalescence and recovery, where we recover. So mechanism by which the disease is caused, the cellular events, reaction, and other pathologic mechanisms occurring in the development of the disease, describe the origin and development of the disease and whether it is acute, chronic, or recurrent infection. There is a multifactorial process, depends on the immune status of the host, the nature of the species or strain, the virulence and pathogenicity, and the infectious dose in the initial exposure. So this is the process of infection. So the organism enter the host, it attaches to the host, or we call it adherence. Then they cause invasion, and then they have evasion. They resist the host defense, the host physical barrier, immune system, adaptation, food sources, and metabolism. They colonize the host. They establish colonies and cause disease. And then they multiply and spread to others. And then they enter the other new hosts, and the cycle continues. So this is the component of infection. There's a portal of entry, the invasion. They attach to the host, they escape or resist the host immunity, and they cause damage. So the portal of entry can be skin, mucous membrane, or parenteral. The skin, unbroken skin provide good protection. Some pathogen gain access through hair follicles and sweat glands. You get the pimples, acne, and some like pathogenic leptospira species, you have a wound, your skin has a cut or wound, so the pathogen can enter uh, through the skin. And then there are also mucous membrane. So most frequent portal of entry are the sites where mucous membrane meet with the epithelial tissue lining. Like the respiratory tract is an easy and most frequent used site by pathogens like respiratory viruses, Streptococcus pneumonia. They can gain entry through the respiratory tract. And then there are also gastrointestinal tract, common entry site for fecal oral like Salmonella typhi, Shigella. They enter through the water, food, contaminated fingers, and fomites of the environmental surface. You touch and you eat without washing your hand, it can gain entry into your digestive tract. So, but it has to survive the stomach acid, enzyme, and bile. And then there is also genitourinary tract. It can be a portal of entry for sexually transmitted diseases like gonorrhea, syphilis. It can gain entry into the host through the genitourinary tract. And then there are also conjunctiva. Your eyes is a membrane covering the eye and eyelids, but if it can become a portal of entry, like Neisseria gonorrhea also can cause ophthalmia neonatorum. It can be a portal of entry also. And then there is also parenteral route. So pathogens are deposited directly into tissue beneath the skin or mucous membrane and cause injuries through injection, bite, cuts or wounds, surgery, puncture, and splitting due to swelling or drying. Like mosquito, uh, they inject the skin and transmit malaria, dengue, tick bite. Uh, they can transmit also the parasite, Borrelia. Uh, then this is skin, you have cuts and wounds. So this can be a portal of entry for Leptospira. So invasion. So once they gain entry, they do the invasion process, whereby the pathogen enter the host and cell. Higher number of pathogen increases the likelihood of developing disease. So they attach to the host. They call as adherence to the host through their properties, like this tapeworm. They have hooks and suckers. They attach to the intestinal tract using this uh, hook and suckers. And some bacteria like E. coli, they have bacteria and fimbriae. They attach to your urinary tract. So pass urine, they still not removed during passing urine because they hold tight to your urinary tract using this bacteria, fimbriae or flagella. So also some bacteria, they have adhesins or ligands to attach to the host. Some have fimbriae and pili, and some have non-fimbriae adhesins to help them attach to the host. So once they have attached, they have to resist the host immunity. They have to, we call it as evasion. It's a process whereby the pathogen that enter the host penetrate and avoid the host defense system. These properties are controlled or mediated by specific virulence factors, which are products expressed by the pathogen that permit a pathogen to cause diseases involved in every stage of infection process at genetic, molecular, and functional level. So they have properties, and such as, for example, Streptococcus pneumonia, they have capsule. So this capsule is a thick protection, which is common mechanism to avoid phagocytosis. Even they have phagocytosis, you are protected by this protection capsule. And then tuberculosis, they have wax, mycolic acid wax. They have another layer, another layer called as a mycolic acid wax. So even if after the phagocytosis, they survive in the white blood cell. Because of this wax, they can survive in the white blood cell, they can multiply even in white blood cell because of this mycolic acid wax. This is important. And then environment production, enzyme production, sorry. So some bacteria, they produce leukocidins. So these bacteria which produce leukocidins, they destroy white blood cells. And some, they, like just now I said, the TB, Mycobacterium tuberculosis, they survive in the macrophage because of the wax. And some have strong cuticles, like the tapeworm. They have strong cuticles. They survive in the digestive tract. And some cause damage to the cell and tissue through three mechanisms: through direct damage, through the production of toxin substance, and some through immunopathology. They cause inflammation, host response, and autoimmune response. So most damage is carried out by the toxic substance they produce. Some bacteria can induce cells to engulf them, such as E. coli, Shigella, Salmonella, and Neisseria gonorrhea. They induce the cells to engulf them. Like Salmonella, they can survive intracellular in Neisseria gonorrhea. They are obligate intracellular Neisseria gonorrhea. So they induce the cells to engulf them. And some metabolism and multiplication kills the host cell, so they multiply in the cell and the cell explode. And others pathogen enter the cell by excreting enzyme or through their own motility. They have flagella to move. Some pathogen, they produce enzymes. So mode of transmission. When we talk about mode of transmission, we talk about a few. We don't have to talk a lot. We just say a few words like fecal oral route, or respiratory route, or sexually transmitted disease. So when we ask what is the mode of transmission, we just use these few words. You don't have to talk a lot. So what is the mode of transmission for Salmonella or Shigella? We say fecal oral route. What is the mode of transmission for COVID-19? What is the mode of transmission for Streptococcus pneumonia? We say respiratory route. When we talk about syphilis, gonorrhea, what is the mode of transmission? We say sexually transmitted diseases. We don't have to talk a lot. Just use this istilah words. So you can answer good. So parasites change some mode of transmission. They change the behavior of the host, like Toxoplasma gondii. It causes the cat it causes the mouse, which is usually afraid of the cat, but it become attracted to the cat. Change the behavior of the mouse. This parasite, when it's inside the mouse, it change the behavior of the mouse. It become attracted to the cat. change of behavior. And then this worm change behavior of the this insect amphibot, aquatic amphibot. So when the worm infect this amphibot, so the amphibot swim towards the light. So they change behavior of the amphibot. It swims towards the light and become closer to the predator, most likely the duck. So also some parasite they exploit their host direction, like Plasmodium species use heat during high fever stage to establish some of its biological process. Plasmodium species causing malaria, so they use the heat during the high fever to establish some of its biological process. And then this pinworm, Enterobius vermicularius, they deposit the egg around the anal area, because when they deposit the egg around the anal area, the host will start itching and he will garu-garu, and then the parasite will be in his fingernail, and when he eat doesn't wash hand, and the life cycle continues. So this is how the behavior changed because of the parasite. So this is my references, and thank you. If you have any question.